Howdy and welcome to the last video of your unit on protein function. We're still talking about hemoglobin and allosterism. While the Bohr effect was relating the proton concentration, or the pH, to the release of oxygen in the tissue and the uptake of oxygen in the lung artery interface, another allosteric effector of hemoglobin is 2,3-bisphosphoglycerate, or 2,3-BPG to you and me. And 2,3-BPG binds to the center of the tetramer of hemoglobin. So remember in that first slide when we first showed you how the the space in the center of the tetramer is wider in the T state and closes down in the R state. Well, it's the T state that binds to the allosteric effector 2,3-BPG, and that means that when this effector binds, it converts oxyhemoglobin, the R state, to the T state and stabilizes hemoglobin in the T state. And so its binding also helps the release of oxygen in the tissues where it needs to be delivered. Okay, so 2,3-BPG binds at a site distant from the heme iron where the oxygen binds, the heme iron that is, and therefore it's called the allosteric effector. There's your Greek allo, other, steric structure. And in fact, it's the presence of the 2,3-BPG that helps that nudging of subunits as one subunit binds that first oxygen molecule, changes its conformation when that oxygen is the sixth ligand to that heme iron, which pulls the heme iron into the plane of the heme, pulling on that F helix, and because of the histidine that's part of that helix, as the fifth ligand. Remember that? Well, all of that movement is coordinated through 2,3-BPG. All right, so in the absence of 2,3-BPG, the oxygen binding would not be sigmoidal. It would be more like the myoglobin oxygen binding curve. It would look more hyperbolic, right, and less S-shaped. All right, so let's have a look at this. This is the structure of 2,3-BPG, and what you notice right away, especially when you look at the central geometric projection of the structure, look at all of these negative charges, okay? So this is a highly negatively charged molecule, and here it is in ball and stick. So you're looking at all of these O minuses, and they are going to be coordinated with plus charges in the center of the deoxy state of hemoglobin. So let's have a look at where BPG binds to hemoglobin. Where does it bind? It binds inside. One BPG molecule binds per tetramer. Okay, this is very different from oxygen. That's 4 O2 molecules per tetramer because there are 4 hemes per tetramer, right? But there's only one central cavity formed by all 4 of the subunits, and that central cavity is where BPG binds. The negative charges that you saw on the last slide interact with 8 positive charges that are inside the cavity, that line the inside of that cavity. 2 from lysines, 4 from histidines, and 2 are N-termini. Now, when you look at fetal hemoglobin, this is the hemoglobin that is in the fetus when it's still inside the mother's womb. The fetus has to win in the competition for oxygen with the mother's tissues, right? So it turns out that the fetal circulatory system is separate from the maternal circulatory system, but the capillaries meet for gas exchange in the placenta. So the placental tissue is going to facilitate the transfer of oxygen from the hemoglobin in that same tissue, and the hemoglobin in the mother's cell, the adult hemoglobin, in toward the fetal hemoglobin. So how did we get that directional transfer from, because we're in the tissues, we're at the point where the hemoglobin is going to be releasing oxygen. So why doesn't the fetal hemoglobin also release oxygen? It's going to be binding more tightly to oxygen than the mother's because it has a lower affinity for BPG. See what the reason that we went around this cul-de-sac to fetal hemoglobin has to do with the allosteric effector 2,3-BPG. Fetal hemoglobin has a very low affinity. It's a tetramer, but it has different subunits. So fetal hemoglobin, or HbF, has the 2 alpha subunits, but instead of the beta subunits, it has gamma subunits. And those gamma subunits are missing some of these positively charged amino acids strategically placed by the beta subunits at that central cavity where BPG is bound, okay? And so without the BPG bound, or if the low affinity for BPG, the fetal hemoglobin has a higher affinity for oxygen. And that's how it wins at the tissue capillary interface of the placenta. It wins the oxygen from the mother's hemoglobin. So here's the BPG circled in the center, and this is the T state of hemoglobin with 2 of the beta subunits in color, and the alpha subunits are in the back in white. And what you see is the side chains that contribute these positive charges, those nitrogens that are carrying those positive charges are dark blue, and they're facing the lining of this cavity in the center of the tetramer. And that is what actually stabilizes the BPG. This stabilizes the T state. So BPG is helping to stabilize the deoxy state of hemoglobin, again helping to shift that transition from oxy to deoxy right where it's needed at the tissue capillary interface, making that release of oxygen much more cooperative and sigmoidal shape of that binding curve.